Welcome to the podcast for Healing Neurology, which is where we talk about everything that can help heal your neurology, which is really everything from food, lifestyle, and medicine to nature, culture, and politics. There's no topic too big or too small. I'm Jillian Ehrlich, family nurse practitioner, certified in Ayurveda and functional medicine. And we have a pretty fun guest here for you today. Dr. Tarun Singhal is Director of PET Imaging Program in Neurologic Diseases at the Ann Romney Center for Neurologic Diseases at Brigham and Women's Hospital in Boston, Massachusetts. And he's also an assistant professor of neurology at Harvard Medical School. Tarun completed his medical school at All India Institute of Medical Sciences and completed a nuclear medicine residency in India prior to coming to the U.S. in 2006. He completed a postdoctoral fellowship at the Yale PET Center and Kettering PET Center, followed by neurology and nuclear medicine residencies and fellowship at Harvard. He is board certified in neurology and nuclear medicine and on the Harvard faculty since 2014. Tarun has led several molecular imaging projects in the neuroscience field, including work on C11 methionine PET in brain tumors, norepinephrine transporter imaging of the central nervous system, and now more recently, neuroinflammation PET imaging in multiple sclerosis and related disorders. And that's what we're going to talk about today. Tarun maintains an active clinical practice of seeing MS patients in addition to leading the recently founded PET imaging program in neurologic diseases at the Ann Romney Center. Oh, what a powerhouse and a very humble spirit. We welcome you today.
Well, I'm so excited to be here. Thanks so much, Jillian, for having me on this show.
Do you do a lot of podcasts?
No. This is my first. I actually have started listening to some, it's a Navy SEAL, Jocko Willink. He has a Jocko podcast. So I've started listening to it. And that's the main one that I listen to. Yeah. But I've never been in a podcast before.
So we were just talking a little bit before the show. I actually think it's really important for people like yourself to be on podcasts because the work that you do is actually really revolutionary. You're really right on the cutting edge of understanding these diseases, and especially with MS. And we had a previous show with Dr. Weiner, and we're talking about how, before we had MRI, MS, multiple sclerosis, was thought to be sort of like a hysterical disease of womanhood. It was anxiety and depression, and we couldn't really start treating it and even taking it seriously until we had the imaging to understand it. And so the work that you're doing can be really critical to how we move forward in both understanding, assessing, and treating these conditions, these neuroimmune conditions.
Yeah, indeed. It's very important to know the target and to visualize it. As Sun Tzu said in The Art of War, if you know yourself and you know your enemy, then you will win 1,000 battles or something to that effect. So if you don't know where your enemy is or how active your enemy is, then, you can't really target it. So same with these neurological diseases. There is this big arm of the immune system with these cells called the so-called microglial cells, which are active in many neurological diseases, particularly multiple sclerosis and others, such as dementias, but are not trackable with the current imaging techniques. And it's very crucial to visualize them and to quantify them and to target them in order to treat these diseases. And there's an emerging understanding in the field that, that's probably a crucial missing link in our approach to successfully treating many of these disorders.
Amazing. So let's start with kind of basics, just so we have building blocks, everybody on the same page. What are microglia cells?
Right. So, in the brain, there are many types of cells. The most popular or the most common ones that people are aware of are the so-called neurons or the nerve cells, right? And but in addition to the neurons, there are these other cells which are supporting the neurons, and they are called the glial cells. So glia basically refers to the fact that initially they were thought of as just glue-like cells. They were thought to be just cells that were gluing together the neurons. And microglia were called as such because they were the small glue-like cells. So micro means small. So these were among the various types of glial cells or the glue-like cells. there were some larger cells and there were smaller cells. So the larger cells were called macroglia and the smaller cells were called microglia. And macroglia are of further two types. One is called astrocytes because they're star-like. And then there is something called oligodendrocytes, which basically produces the insulation around the nerve cells, the myelin sheath, so to speak. So microglia were identified as such in the early 20th century. And over the last 100 years or so, and more so in the last 2 decades, it has become clear that they are more than just glue-like cells. They have lots of functions in the brain, both in health and in disease. So they support normal functioning of the nerve cells. And they also make sure that the development of these nerve cells follows the right pathway when even as early as one is in the womb, as a fetus or embryo, the microglia cells have a role in the normal development of the brain and the central nervous system. And subsequently, in health, they support the nerve cells. They track the territory around the nerve cells in the brain for any intruders, any abnormalities, take care of any debris, like any other issues that are going on. They're basically kind of the gatekeepers or sort of the protectors of the brain. In disease conditions, these cells can, however, become abnormally activated. And then they start producing potentially harmful chemicals or start even eating up nerve cells. In some ways, you can say that they can become misdirected. So that's their relevance in the normal brain functioning in health and in disease.
When they were thought of as glue, it seems like it was really like all of their communication. They had not tendrils like an astrocyte, but really that gluey piece was really all of that cellular communication going to the different areas. Is that right?
Right. So, in their healthy state, they have the so-called ramified appearance, which means they have a lot of sort of tentacles around their central core, which basically scans the territory in which the nerve cells are residing, which are in a given cell's territory. And when they are abnormally activated by any issue that is going on in their vicinity, then they start changing their shape. So instead of having those long sort of ramified these tentacles, they start becoming more and more round and globular. And in that process, they also express newer molecules on their surface, which are responsible for interacting with other cells, with the nerve cells, other immune cells, the nerve cells, and also with any debris, etc., in the environment. And they also then start secreting these harmful chemicals that can cause problems. They can also be protective to the brain, even in these disease conditions, because they can secrete some so-called trophic chemicals, or the ones that provide nourishment to the nerves. So they have like sort of a kind of a dual role in that sense, like Dr. Jekyll and Mr. Hyde sort of, potentiality. And that's what scientists are trying to understand more and more, how in various disease conditions and at different stages of the same disease, these cells change their functioning and contribute to either disease progression or preventing injury.
Do we have a sense of why that shape changes in that way? Like, what is the benefit to the cell or the benefit to the brain to go from those tendrils out to kind of that globular structure?
Yeah, I think from the so-called teleological perspective, or like what is the actual functional benefit to the brain, one can say that earlier their goal was to overall scan the territory. So they wanted to, just look at many cells. But when they changed the shape, they have become more focused on the target that they have identified, in a globular sense, and then they can either secrete the chemicals, the cytokines, so to speak. Those chemicals are called cytokines, or can engulf more and more of debris in their area. So therefore, they tend to become more focused and globular. That is one way of thinking of that.
And so microglia are just in the central nervous system. Is that correct?
That's true. There are cells like that in other organs, but they are not called microglia. They have different names and also different origins in terms of where they started in the embryo, etc., and also different cellular and molecular makeup. But many organs, including the liver, for example, have such sort of intervening cells that lie in between the primary cells of that organ that kind of have these functions of supporting and protecting those cells.
So we're acting like the healthy microglia right now. We're making these connections. We're having this communication.
Exactly. And providing nourishment to the neurons of all of us,
Exactly. That's exactly right.
Right.
So inside the central nervous system, if there's a trigger, how localized is that trigger versus, if there's a trigger in one area of the brain, do all of the microglia become that globular structure or kind of activate or shift? Or is it more like different parts of inside different parts of our brain, there's a constant hum of, like sensing or responding or sensing or responding?
Yes. So I think it's both. So there is a constant hum in all parts of the brain. In some parts, it's more than others. So in terms of the density of these cells, there is a variability in different regions of the brain. So like, for example, one of the regions of the brain is called the so-called substantia nigra, because it looks dark or black when you look at it in pathology specimens. So it's a dark or the black substance, substantia nigra, which has a high density of microglia for unknown reasons, at least to me. So there's a variation in the number of microglia that are there in various parts of the brain. But if there is an injury in one part of the brain, there is evidence that microglia can migrate from another region of the brain to help their colleagues.
Oh, wow.
In that region. Yes.
They come over.
Yeah, it's, the army analogy or the, armed forces analogy is very much applicable here, So in health, they are really the protectors of the, whatever the country or the city or, whatever area they're protecting. But in disease conditions, they can become misdirected. That's the problem.
So one more little question that I'm wondering, and so some of our listeners might also be wondering, I'm wondering if you can run through some of the names of those molecules, the cytokines or chemokines or hormones or neurotransmitters. Like, what are some of the chemicals that these cells use in both their sensing and their communicating?
Yeah, so, they will sense anything that they think is potentially problematic. And they have molecules on their surface called DAMPs or PAMPs, which are basically disease-associated molecular patterns, which they will sense through various receptors on their surface, including so-called toll-like receptors or TLRs. Then in response, they can produce various cytokines, such as those called interleukins, which have various numbers. So, they can interleukin 1, interleukin 6, so IL-1, IL-6, etc. They can produce TNF alpha, which is like a tumor necrosis factor alpha. They produce a lot of oxygen-free radicals, which can cause oxidative injury to the nerve cells. So there are a huge list of such chemicals that are there. There is a class of molecules called chemokine attractant proteins, CXCL3, etc. So there are a lot of those.
So lots of communication. When we think about our bodies, I think we think about our structure, like we think about our bones, our muscles, our skin. I think recent science has started really looking at our microbiome. So now we know that there's, billions of microorganisms inside of us. And so now we can add to that list of what's contained within our borders, also all of this communication. Like, this is a real hum. There's a real cacophony of all this communication going on, eh?
Yeah. And, among the receptors on these cells, you should also think about receptors for the conventional neurochemistry molecules, such as norepinephrine, serotonin, dopamine, etc. So, for example, one of the things we are studying is the interaction between norepinephrine and microglia. So norepinephrine or noradrenaline, people would be familiar with adrenaline, right? The adrenaline rush that we have in the body when we have stress, anxiety, etc., fight or flight response. There is the central nervous system equivalent of that is called norepinephrine or noradrenaline. And that is supposed to have an anti-inflammatory effect on these microglial cells. So not only do microglial cells affect the nerve cells by these various mechanisms that we talked about, but the nerve cells can in turn affect the microglial cells. And these effects may be mediated by these neurochemicals such as norepinephrine. And there is evidence that degeneration of nerve cells that produce chemicals like norepinephrine are associated with higher microglial activation and may contribute to propagation of disease faster and to a more serious level. So for example, in Alzheimer's disease, one of the regions of the brain called the locus ceruleus, or the so-called blue spot, ceruleus means blue. So that locus ceruleus is a main site of nerve cells that produce norepinephrine, and it has widespread projections throughout the brain. And in Alzheimer's disease, this locus ceruleus undergoes early degeneration. And it has been shown that in animal models, at least, that the degeneration of the locus ceruleus is associated with an increased inflammatory response in the rest of the brain and increased microglial activation, which then helps propagate the disease faster and further in those subjects. So at least in those animal models. And now further research is being done in humans to see this, look at this crosstalk between the nerves and the microglia in the reverse direction as well. So this could have a role in like stress-induced inflammation and can be a potential therapeutic target.
So when we talk about this family of neurodegenerative diseases, Alzheimer's, Parkinson's, the microglia are involved, it seems like, in all of them. Is that correct?
Yes. So there is clear evidence that microglial activation is present in humans who have these diseases: Alzheimer's disease, Parkinson's disease, multiple sclerosis, amyotrophic lateral sclerosis, or ALS. It's well established. So it's known not only from pathological literature where, either biopsy specimens from patients' brains or patients have passed away, then the autopsy specimens are looked at and there is clearly activation of microglial cells. But now, in this day and age, you can actually look at these microglial cells with an imaging technique called PET imaging, or positron emission tomography, where basically a small amount of a dye is injected, which goes and binds to these activated microglial cells and can then be used to image where and how much these cells are activated. And, it's a fascinating technique, the PET imaging of microglia, and has provided us a lot of novel insights into this.
Talk to us a little bit more about PET.
Okay.
Yeah,
sure. So I love this topic because, PET imaging is really a unique type of imaging in biomedicine, which brings physics, chemistry, and biology together in real time to provide novel insights at a molecular and cellular level. So PET stands for positron emission tomography. So positron is basically a subatomic particle. So, in the atom, there is the atomic nucleus and there is the electrons that are there in the orbit. And the atomic nucleus has protons and neutrons, and there are electrons that are around it, right? So the electrons are negatively charged and the protons are positively charged, right? So positron is basically a positively charged electron. So it's an electron particle, but it's not negatively charged, unlike the normal electron that we have in the world, in the universe, I should say, but is actually positively charged. So it's in the physics sense, it's actually a type of antimatter to the matter, which is the electron. So this, we're talking a little bit sci-fi, like, quantum physics, particle physics now from biology. But that's what I'm saying, that's why it's so fascinating to me, because it combines physics, chemistry, and biology in real time. So let me just go into a little bit of detail, which is not complicated at all. Because you see, positron, as I said, is an antimatter particle to the matter particle, that is the electron. All matter particles have a corresponding antimatter particle. And it is not only believed, this is a theory according to the Big Bang theory, that when the universe started, within the first 10 raised to power minus 36 seconds of the Big Bang, the antimatter particles disappeared. And nobody knows what happened to those particles. And that phenomena is called baryonic asymmetry, or baryonic, B-A-R-Y-O-N-I-C, baryonic asymmetry. People might want to look it up. So these antimatter particles disappeared. So they don't exist in nature for the most part. So the entire universe that we have is actually made of matter particles. We don't have antimatter particles per se necessarily for the most part. We do get some antimatter particles in natural conditions, for example, in storms and hurricanes. And people have found that antimatter particles such as a positron is created in a storm because of the high energy and the interaction among the particles there, that there are some atomic changes or subatomic changes then lead to production of these positrons, which have been, people, how they fly into these storms sometimes, to study them. And they have detected these rays that are coming from these antimatter particles or positrons. But obviously, when we are imaging them in the hospital, we can't create a storm every time we do that, right? But except that we can do that in a controlled device called a cyclotron, a medical cyclotron. And I'll talk to you about that in a second. But so coming back to the positron. So positron is this positively charged electron. It's an antimatter particle. And in PET imaging, we use isotopes, radioisotopes that are basically isotopes of the naturally occurring elements, but have this property that they can produce a positron. And when they produce a positron, which is an antimatter particle, what happens is that this antimatter particle, because it's really very unstable, it is not a natural, so to speak, naturally stable particle. It interacts with the electrons which are there all around it. And it is the inherent property of matter and antimatter particles that they don't like each other. And they annihilate each other. So their mass gets converted into energy. And that energy is then emitted as gamma photons or gamma rays, which are then picked up by a PET scanner and then converted into an image. And that's why the term positron emission tomography, because it depends on radiation being emitted from the interaction of the positrons with electrons, as opposed to a CAT scan, conventional CAT scan, where the source of radiation is outside the body and the energy is transmitted through the body. Here, the energy is emitted from the body because a small amount of a radioisotope has been injected into the body to target a particular biological process. And then tomography basically means imaging by slices. So that's tomos, means slices. So that's positron emission tomography. So that's PET scanning. So, and then for microglial PET imaging, we use isotopes that produce positrons and tag them onto some molecules that will go and bind to these microglial cells. And in particular, these days, the molecule that we target is called TSPO, or translocator protein. It is present in these microglial cells, in the so-called mitochondria, within those cells. And when there is increased number of such microglial cells in the brain tissue, then they have more TSPO, or this target in the brain tissue. So when you inject a PET radio tracer, which has this positron isotope tagged to a molecule that is known to bind to TSPO, it goes and binds to the TSPO. The more molecules of TSPO there are, based on the amount of microglial activation, so to speak, the more these PET isotopes, PET radio tracer molecules will bind. There will be more radioactive decay, more energy will come out of them, more signal will be picked up by the PET scanner, which will then identify where exactly it came from and convert it into an image for us to see. That's how this works.
Wow. You make it very simple.
This is 5 decades or more of work, scientific work that, as we say, right, we are able to see far because we stand on the shoulders of giants. So this is decades and thousands of scientists who have made it possible for us to even understand it at this level.
Can a PET differentiate between what state the microglia is in then? So whether it's got its tendrils out or it's in its globular form?
That's a great question. That's a wonderful question. So the ultimate goal is to identify the exact state of microglia. And right now, the tracers that we have, they look at the so-called microglial density. So it tells you about the number of microglia that are there in the tissue. And most often, that density has been associated with an abnormal activation, that it is associated with injury to the underlying brain tissue. However, there are some situations where there can be an increased density, but that increased density is occurring to protect the brain cells. For example, in earlier stages of Alzheimer's, there may be an increased density of these cells to engulf the abnormal amyloid protein that is deposited in Alzheimer's. So therefore, right now, the TSPO target basically tells us about the density, but cannot 100% distinguish between the pro-inflammatory versus the anti-inflammatory subtype of microglia. And that is work that is ongoing to identify newer molecules that need to be targeted, because you see, PET imaging is very versatile in that sense. You have a target that you know is of relevance, you can then design molecules and tag them with these PET isotopes to target those molecules. So you can have different, so every PET scan is not the same, right? So there is this glucose PET scan that people have talked about as used in other conditions like cancer and cardiac issues, and sometimes even in the heart, in the brain, I'm sorry. But it just depends on what molecule you're injecting. So if it's a radio-labeled, glucose molecule, then you get a glucose metabolic study. But then if it's another molecule, so you can target different molecules going forward. And there are many such PET radiopharmaceuticals in development right now to look at these exact subtypes. And it's also confounded by the fact that the microglial biology itself is being understood better on a daily basis. So there is a technique called single cell RNA sequencing, or so-called RNA-seq, which is now telling us that there are many subtypes of microglia, not just two, like pro-inflammatory and anti-inflammatory. That's a oversimplification. And then, as that biological understanding evolves, the imaging molecules will also evolve over the next so many years, or to next couple of decades, to kind of enhance that differentiating ability.
One of the most common complaints in conditions like this would be something, gets called by patients brain fog. What do you think of brain fog?
Yeah, so, our own research, we have found that fatigue, for example, in MS patients, which is known to be like a major symptom in MS, in multiple sclerosis, 80% of patients complain of fatigue. And in vast majority of patients, it's a leading cause of disability, in terms of cognitive fatigue, physical fatigue, psychosocial fatigue. And we found a strong correlation of fatigue scores in MS patients with microglial activation using PET. Particularly in the region of the brain that I mentioned earlier, the substantia nigra, which is a seat of dopaminergic neurons in the brain. And also in many other areas of the brain that are relevant for cognitive processing, alertness, attention, and mood and energy levels, such as the so-called precuneus region of the brain, which is a hub for a network called the default mode network. So the abnormal microglial activation was found in very relevant areas of the brain to be strongly correlating with the fatigue scores in MS patients. So that is from our own work. And others have also shown that in a condition called chronic fatigue syndrome, there is increased microglial activation seen with another PET molecule that targets the microglial activation and correlates with the severity of symptoms in even chronic fatigue syndrome.
Do we think that this microglial activation in these conditions is a cause or a result?
Right. So that's a great question. Obviously, previously it was thought that it may be all just reactionary and therefore maybe a result of some underlying, whatever the underlying illness is. But now there is increasing recognition that this abnormal microglial activation can accelerate the disease process, and in some conditions can play a key role in even initiating it and propagating it. So in other words, it is probably both ways. It can be both a cause and a result. But there is more and more recognition that it is potentially contributing to the worsening of these diseases for, at least, other than the fact that it may be actually causing the disability in some of the conditions.
My Ayurvedic teacher, Dr. Ladd, has said before about pain that initially pain is a response, to alert you. It's a response. But then if the pain is long enough, deep enough, intense enough, then it becomes its own disease. Exactly.
And that's a great insight. And interestingly, PET imaging has shown that there is increased microglial activation in chronic pain patients. So, and then it becomes a self-propagating cycle in that sense. And that can, contribute to underlying neuronal injury, and then leads to abnormal processing of whatever sensory inputs one may be having in the brain. And then, this propagation of the symptoms such that they become refractory. So I think what they're saying makes total sense that, once there's persistent of this type of and inducing pathology, in this case, pain, that itself causes some kind of an injury, which becomes somewhat permanent and mediated by the microglial activation.
Do we think that this is part of TBI also, traumatic brain injury?
Yes, there is clear evidence that there is increased microglial activation, sometimes even years after traumatic brain injury. And even on PET scan done years later in patients who had initial traumatic brain injury, there is abnormal microglial activation.
Oh, wow.
And now in animal models, that is being targeted with newer therapies, not just in TBI, in all these diseases, to kind of see how we can slow these diseases or even to hopefully reverse some of the changes.
So just a few more questions about different diseases, and then we'll move into kind of therapies and treatment options. But what about mental health conditions, anxiety, depression? What about OCD or ADD, ADHD, attention deficit hyperactivity?
So in depression, there are multiple studies using PET imaging that have shown abnormal microglial activation in depression patients, particularly in the regions of the brain that are known to be involved in depression pathophysiology. In our own work in MS patients, we have seen that there are certain regions of the brain that have increased microglial activation, such as anterior cingulate, which is a region that is responsible for motivation and is known to have low glucose uptake on the glucose PET scan. But on our inflammation PET scan or the microglial PET scan, we see that there is increased microglial activation, which actually goes hand in hand with the fact that there is reduced neuronal activity and low glucose in the same area. And then there is another region of the brain, so-called subgenual cingulate, which is another part of the cingulate gyrus. Cingulate basically means the belt. So it's like a kind of a belt going from front to back. So there are different parts of this cingulate gyrus. Is anterior cingulate, posterior cingulate. Then there is a subgenual cingulate because it is below the knee of the corpus callosum. Genu means knee. So all these terminologies, the etymology is fascinating. So the subgenual cingulate has been shown to be overactive on glucose PET scans. But then when we look at the microglia, we see actually a decreased signal on our preliminary work. So there may be the component of glial toxicity there, like glia are themselves getting injured. So, and then others have shown that in MS patients in depression, there is increased inflammation in hippocampus. But even in just idiopathic, like major depression, which is independent of MS and other diseases, there is a lot of evidence for abnormal microglial activation in these or very similar areas of the brain. As far as the other conditions, ADHD and ADD, etc., I'm not particularly sure. I'll have to look up the literature on that. Bipolar disease, there is some evidence about the role of inflammation, and OCD also. So this is a very active area of investigation in neuropsychiatric conditions. And as I said, in several of these, there is already a lot of evidence.
And we know that the PET, this very specialized, this is not something you can go to your doctor, your primary care provider, your GP and say, hey, can I get a microglial PET scan, please?
Yes. No, it's not. It's not. It's a research tool right now. And hopefully with more and more data accumulating, we will reach a point where there is a molecule that is approved by FDA for routine clinical use, depending on the scenario, the context, and the treatment options that are available. But you mentioned Dr. Weiner, so he always likes to say that, like MRI revolutionized the care of MS, X-rays, chest X-rays revolutionized the care of TB. So, tuberculosis, so imaging has a very important role. And therefore, we need to keep developing these newer tools for these neurodegenerative, neuropsychiatric conditions where really we don't fully understand what exactly is going on. And the conventional imaging techniques like MRIs may actually be normal or remain unchanged while the patients continue to get worse. So definitely down the line, there is a lot of scope for these approaches to become more mainstream and clinically available.
Do we have are there currently available labs that can reflect microglial activation or neuronal impact? I know kappa and lambda light chains is one or light filament Can you talk a little bit
Right.
about that?
Yeah,
Or
neurofilament light chain. Yes, NFL.
neurofilament light chain. That's
Right,
what I meant.
right, exactly. Yeah, that one. So yeah, that is a marker of neuronal injury, mostly attributed to the axonal injury. So the nerve cells have the cell body and the axons, which is like the wire. So if there's injury to the wire or the axon, then the NFL tends to come out. But overall, it's considered a marker of neuronal injury. And now it's actually available test that one can get it ordered, in the clinical setting. Insurance coverage may vary, depending on, that's always a concern. But yes, so that is a marker of neuronal injury. And like in MS, for example, when there is relapses, there is sudden increases in those levels. But, it's not foolproof. Like it's not definitely shown to be 100% predictive either way of, a good outcome or a bad outcome, but it can be helpful. Mm-hmm. Similarly, there is another serum marker that is emerging. It's called glial fibrillary acid protein, or GFAP. Mm-hmm. It's a marker that is secreted from astrocytes, the star-like cells that I was saying, this kind of the macroglia. For the microglia per se, there is no specific molecule, although some are being looked at in the context of monocytes, which are these circulating sort of cells that can become macrophages when they go into the tissue, like, they become those cells that engulf the debris. So, among the blood cells, the white blood cells in the blood, there are these neutrophils, lymphocytes, monocytes, basophils, eosinophils, right? So monocytes is a type of white blood cell, different from the lymphocytes. So there are some markers of monocytes, such as MCP1 and MIP, etc., that are being looked at, but they're not like they cannot be considered as microglial markers. They're more like markers of monocytic function. And another fascinating thing in research is to look at the interaction of these microglia cells with these peripheral cells, these are monocytes and T cells, and so that's another active area of work. In terms of other blood measurements, one can do, again, this is all in the research realm, not available clinically. One can do single cell RNA sequencing of the blood cells and see if there are changes there. Mm-hmm. So these are all things that will potentially become available for clinical use in the next few years. But right now they're in the realm of research. That's why the imaging becomes even more relevant, because there's no other way of knowing what is going on in the brain, because you see the brain is protected by the so-called blood-brain barrier, and not much that goes on in the brain is necessarily showing up in the blood. So that's why sometimes you have to do the spinal taps, or the so-called number punchers, to get cerebrospinal fluid to get a better idea of what is happening in the brain.
What are the specific markers in cerebrospinal fluid from lumbar puncture that might reflect?
Not really. I'm sure there are many that, there are some that are being looked at by different groups, but there's nothing one that is like a consensus molecule that we say, oh yeah, this is a marker of microglial activation. In fact, I was presenting at a recent national meeting, and then we were talking about how do you validate these PET markers in humans, and we were talking about how in amyloid PET imaging, the PET was validated against autopsy specimens in Alzheimer's patients. But in MS, for example, that is not the scenario often, because our patients are younger, and you don't usually image them in the last year of their life, as opposed to in Alzheimer's disease, that is what people do often, and especially if you're doing a research study. So somebody said, well, we could potentially do a CSF marker, and then people ask, well, which one? But because there's none as such. I mean, there are many in the research realm, we can talk about them, but then there's none that is validated enough to say, oh yes. Because then if that had been the case, we would have said, okay, that's the one we are going to use. And then PET can just show us the regional variation in the microglia, while the CSF can show you the overall picture. But that's not the case at all.
We know a lot about these microglial cells. We know quite a bit about their role in neuroinflammation. We can see them so far just kind of with PET. We know that there's sort of a very common response for neuroinflammatory conditions. So here's the really the $50,000 question, the $5 million, the $50 million question. I don't know how much money you can put to this, but what do we do about it? That's the question. How can we do microglial anti-inflammation work?
Exactly. So, I mean, I don't know how much money you can put the money value, because, it could be like hundreds of billions of dollars, depending on, right. But then most importantly, it's about patience. It's about, human suffering, and can we reduce that? And that is priceless.
Yeah.
So, and that's what we are most driven by. We don't want to image something just for the sake of imaging. I mean, obviously we want to understand the disease and everything and kind of get better insights into the pathological process. But ultimately, as a physician, as somebody who sees patients with these conditions, particularly multiple sclerosis and others, I want to do something about these problems and treat it. So we are very active in researching novel treatment approaches that target microglial activation. And we are looking at the effects of the current therapies on microglial activation to see, what is possible with the therapies that are already being used, for example, in MS. But we know, for example, a subset of MS patients are the so-called progressive MS patients, and they, continue to decline. Their MRIs remain unchanged, but clinically we know that they are getting worse. And we have shown that there is an increased microglial activation in those patients as compared to even the so-called relapsing MS patients. So there's no therapy that is approved for the so-called inactive progressive MS, which are, progressing but are not showing active inflammation on the MRIs or relapses. So, we are looking at novel treatments that can be nasally administered to kind of calm this inflammation down. There are other therapies that are being looked at by various drug companies in terms of oral medicines that target a molecule called the Bruton's tyrosine kinase. They call the Bruton's tyrosine kinase inhibitors, or BTKIs. And they're supposed to help with reducing various lines of inflammation, including potentially microglial activation. So that's the theory anyway, based on the animal work that has been done in these. So there are many molecules in the pipeline. There is one that affects the so-called CSF1R receptor, which is there on the microglia cells. And there are others that, an old antibiotic called minocycline, which has been used for acne, has been shown to have an effect on microglia in some animal models and even in some human studies. In fact, one study, again, because a lot of my work is in MS, in the MS population, it showed a study in patients, it showed that it delayed their conversion to full-blown MS after their first attack. But ultimately, the number of people who converted was the same, but it delayed it. So it had some effect potentially. That was one study. But then in some other disease conditions, it may have made the disease worse. So like people are, again, because you see, it's the yin and the yang, Dr. Jekyll, Mr. Hyde of the microglia in that sense. You don't want to inhibit them in the wrong way if they're actually performing beneficial function. So it's not just about, okay, get rid of these, because they may be performing some beneficial function. You want to get rid of the bad aspects of the microglia. So it has to be done in a very sophisticated way by sort of a very comprehensive understanding of the immune system and its interaction with the nervous system, such that you're not just blindly inhibiting the microglial cells, but doing so in a targeted fashion, such that it affects their pro-inflammatory sort of phenotype or pro-inflammatory aspects rather than their beneficial aspects.
In some of the natural products that we use, so for instance, the herb ashwagandha, very commonly used Ayurvedic herb, or ashwin means horse, and gandha means snort or smell. So it's supposed to give you the strength of a snorting stallion, this ashwagandha. Oh, nice. But then in Latin, it's Withanthia somnifera. And so the somnifera is because it can actually help you sleep. So the question is like, well, how does one herb have this like strengthening as well as this sedating quality? And the idea, that's kind of the range of what we would traditionally call like an adaptogen. So if you're up, it helps bring you down. If you're down, it helps bring you up. And so there's that level of intelligence, kindling intelligence that we want for, out of our herbs, but also as part of our medicines. We don't want to just step on something. We don't want to suppress something, and we don't want to just put something up, we want to help our cells to have greater intelligence, greater discernment, so that those
Exactly.
microglial cells can activate to the appropriate amount for the appropriate duration.
Yeah, that makes so much sense. I think this ancient wisdom has a lot of implications for what we are trying to do with modern medicine and with the modern techniques. Because you see, our bodies are ancient, because the biology has been there forever, in that sense. So, what the ancients knew like a couple of thousand years ago, they were actually working on the same system that has been there for a few 100,000 years, if not millions of years, depending on what you're talking about.
Yes.
So, yeah, we can learn from that approach. I would like to learn more about it.
I'd be happy to work together. Very fun.
Sounds good.
Have there ever been studies or like, I guess it would be like serial PET microglial activation scans on either meditators or people being trained in meditation or people doing breath work, or like say after an Ayurvedic panchakarma and Ayurvedic detoxification? I mean, and part of the reason that I get so interested in those things is that something like breath, there have been a few studies with breath that have shown that breath can influence CSF flow through the brain and through the ventricles. So we know that there are changes that breathing can intentionally create.
Definitely. I mean, clearly there is data that meditation and the deep and slow breathing, they reduce overall inflammation levels in the body, right? There is, I know of many studies that, that target that. I'm not sure about the microglial PET literature there, but that would be very important to do. Another aspect, I think, on the same lines would be exercise. Exercise is known to have significant anti-inflammatory effects. And that has not been studied comprehensively with microglia PET. Irrespective of the PET aspect, I mean, exercise is something that needs to be encouraged, right, for everyone, in general, but particularly for patients who are either at risk or already have a neurological disease or a neurodegenerative disease like the ones that we've been talking about. Because that's one thing I always tell my patients who have MS, that if you are exercising while you can and you have MS, it's like you're taking an additional disease-modifying treatment beyond what you are already doing. So you may be doubling your protection by just actively exercising in addition to taking whatever medicine you are taking. So that the importance of that cannot be, and the importance of exercise cannot be overstated in this context. Parkinson's disease, Alzheimer's disease, there are multiple studies that's been shown that exercise helps these conditions. And one of the mechanisms is potentially through the beneficial effects of exercise on inflammation. And with this exercise, you don't have to worry about if it's going to reduce the good inflammation, because it's a natural way of doing the things that your body should be doing anyway. So it's going to modulate, and the cells will have the intelligence, the one that you were talking about just now, to kind of, do the right thing for the body. But it's just, what I'm just saying is that, from a scientific perspective, it would be good to be able to demonstrate that one of the ways exercise is helping these diseases is by reducing inflammation or reducing microglial activation, for example.
What about the drugs? Like, what about rapamycin or metformin? We know that they have some kind of very interesting longevity impact. And I'm just curious if you think they may have any impact on microglia.
I think they may. The metabolic pathways that are affected by metformin, for example, potentially have a lot of implications for neuroinflammation. And, for example, the entire metabolic state of the body may be modifying the immune system and then getting modified by the immune system. So, and there's so much crosstalk, I think, between the metabolic pathways and the immune system, that again, what we eat, how we modulate our metabolism, and you mentioned gut microbiome at one point, there is a huge crosstalk there, and I think that they can definitely modulate the microglial activation. One particular approach that comes to mind is the so-called ketogenic diet that is used, for refractory epilepsy, for example. And one of the potential mechanisms there is that it reduces inflammation. And again, studies need to be done to see if it's affecting neuroinflammation in the brain itself, which I suspect, that's my suspicion. I mean, I don't have any data that it might.
And so sometimes being in a ketotic state is thought to trigger autophagy, so that it's kind of like a house cleaning. The closets are emptied, the trash is taken out.
There you go. So, the exact pathways, how that happens, obviously needs to be studied, and it's hard to study in humans except with such non-invasive techniques such as, imaging.
Wonderful. Amazing. What an amazing show. You're fantastic.
Well,
thank you so much for joining us today.
Thank you so much for having me. It was a pleasure.
And thank you all for listening today with Dr. Tarun Singhal. We've got lots of ways to continue this conversation through Facebook, Instagram, and Twitter. And you can get more information from and about us at our website, centerforhealingneurology.com. Please be sure to share this show with your friends. We welcome your rating and review wherever you get your podcasts, and feel free to send topic requests to podcast@centerforhealingneurology.com. We love that you've joined us today to discuss how to make our whole world medicine. We rise or fall together, and we're committed to doing what we can to make as many of us as healthy as possible. And this takes all of us, including you. Thank you for listening, and we'll see you next time.
Partyfish Media.
Partyfish Media acknowledges that it operates and records on indigenous Duwamish and Puget Sound Coast Salish land that is still home to the Duwamish tribe. This land is stolen in violation of the Point Elliott Treaty of 1855. We are committed to uplifting the name of these lands and community members from these nations who reside alongside us. For more information on this land, its people, or ways you can help, visit duwamishtribe.org or realrentduwamish.org.